Human reproduction relies on a highly sensitive, top-down chain of command. If the communication network fractures, the entire biological process halts. This control loop is known as the hypothalamic pituitary gonadal, or HPG, axis. It links a specific region of the brain directly to the reproductive organs. When a patient presents with severe pathologies like polycystic ovary syndrome, endometriosis, or hypogonadism, the root issue is often a signaling failure along this exact axis. To understand these conditions, we have to map the baseline neuroendocrine system from first principles, isolate the stressors that break it, and examine how modern medicine attempts to reboot the loop. A reproductive failure rarely begins with a physical defect in the organs themselves. It starts as a systemic breakdown in chemical communication. This schematic maps the baseline communication pathway of the HPG axis, tracking the top-to-bottom sequence from the brain down to the gonads. The cascade begins in the brain. The hypothalamus acts as a pacemaker, releasing pulses of GnRH downward into the anterior pituitary gland. The pituitary receives that signal and bifurcates it. It synthesizes two specific chemical messengers, follicle-stimulating hormone and luteinizing hormone. In males, these signals travel down to the testes to manufacture testosterone and drive the physical assembly of sperm. To prevent biological overdrive, the system monitors itself. As sperm production rises, the testes release a glycoprotein called inhibin. Inhibin travels back up to the brain to suppress FSH, closing a strict negative feedback loop. Returning to this schematic and looking at the left side, we see the female equivalent. Here, FSH stimulates the maturation of ovarian follicles. LH then spikes to trigger the physical rupture of the follicle during ovulation, while regulating the production of estrogen and progesterone. Human fertility depends entirely on the tight quantitative regulation of this biochemical loop, operating flawlessly in both sexes. This flowchart illustrates the primary variables that disrupt healthy hormonal balance. Chronic stress forces the adrenal glands to flood the bloodstream with cortisol, which directly suppresses the brain's synthesis of GnRH. Without GnRH, the downstream production of FSH and LH completely stalls. Endocrine-disrupting chemicals pose another threat. Heavy metals and specific plastics often behave as estrogen mimics. They trick the pituitary gland into registering artificially high hormone levels, causing it to shut down native production. This disruption is compounded by advanced age, specific genetic mutations, and precise nutritional deficits, like a lack of vitamin D or iron, which degrade the raw materials needed for hormone synthesis. The inability to conceive a child is the final physiological symptom, but the mechanical origin of that infertility is almost always located far upstream. We can trace specific diseases to exact failure points in this network. In polycystic ovary syndrome, altered LH receptors and elevated testosterone cause the ovarian follicles to arrest mid-development, building up into cysts instead of releasing an egg. In functional hypothalamic amenorrhea, the physical stress of caloric deficit or extreme exercise cuts the GnRH signal line entirely. Ovarian function drops to zero because the brain simply stops calling for it. The exact same upstream failure occurs in men. When pituitary LH drops chronically low, testosterone production plummets. Without that chemical stimulus, the physical tissue of the testes begins to atrophy and shrink. The sperm count drops below the healthy threshold of 15 million per milliliter. Local structural anomalies like a varicocele, an enlarged vein in the scrotum, can further alter the concentration of the remaining hormones. A cyst on an ovary or a low sperm count presents as a localized physical defect, but treating the organ alone ignores the systemic network error causing the damage. Repairing the loop requires precision diagnostics. Clinicians use blood tests and immunoassays to measure the exact circulating levels of LH, FSH, and estradiol, pinpointing the missing chemical in the chain. If the gonads fail entirely, hormone replacement therapy acts as a direct exogenous patch, supplying synthetic testosterone or estrogen to fulfill the body's baseline requirements. This diagram shows how we kickstart the reproductive cycle when native signals fail. By introducing specific exogenous compounds, like HCG or letrozole, we biochemically force the ovaries to mature a follicle, completely bypassing the hypothalamus. This flowchart outlines in vitro fertilization, the ultimate mechanical bypass of the HPG axis. The protocol requires chemically stimulating the ovaries, surgically retrieving eggs, processing sperm, forcing fertilization in a lab, culturing the embryo, and transferring it into the uterus. These fertility treatments operate as applied systems engineering. We isolate the broken biochemical pathway and deploy specific pharmacological tools to override it. The goal of reproductive medicine is to restore the HPG network to its optimal quantitative thresholds, pulling testosterone levels, GnRH pulses, and sperm counts back into their healthy baseline ranges. To achieve this, researchers are deploying AI-driven diagnostic tools to analyze massive patient datasets. The objective is to map subtle hormonal fluctuations and predict diseases long before clinical symptoms surface. Diagnostics are also moving down to the DNA. By identifying mutations in the FSHR or CYP19A1 genes, clinicians can tailor precise hormone therapies to an individual's unique genetic architecture. Instead of relying purely on synthetic overrides, emerging regenerative fields like stem cell therapy aim to repair the damaged endocrine tissue itself. By decoding the exact mechanics of the neuroendocrine network, reproductive medicine is shifting away from managing end-stage symptoms and towards the precise, proactive engineering of human homeostasis.